SwayamPrabha. Digital India. Educated India.
Welcome back to my course, that Industrial Biotechnology. This antibiotics, Streptomycin, this is produced through the bacterial fermentation process. So, so let us give the introduction to Streptomycin. It is a broad spectrum antibiotics and belonging to the what you call aminoglycoside family. This is very important. This is aminoglycoside family. We know that kanamycin also include in this, gentamicin, kanamycin, that include on this particular category. Then it is active against the gram-negative bacteria, and a number of bacterial infection such as the tuberculosis, Mycobacterium avium complex, endocarditis and brucellosis, the different plague and different rat virus fevers are treated by Streptomycin. The Streptomycin, since it is a broad spectrum antibiotics, it can be used for killing the different germs. Now, if you look at the organisms, that Streptomyces griseus, this is used for the production of Streptomycin, and this is commonly found in the soil, and it is a gram-positive bacteria. The Streptomyces griseus strain are well known producer of antibiotics, and significantly it is a secondary metabolite. I told you that your penicillin also secondary metabolites, also cephalosporin also considered as a secondary metabolites. They produce gray spores, masses, and gray yellow reverse pigment when they grow as colony. They grow at the white pH 5 to 11. This is the specialty of this organism, and this is how they looks under the microscope. If you look at the history of Streptomycin, it was first isolated October 19, 1943 by Albert Schutz, the PhD student laboratory in Selman Abraham Waxman at Rutgers University, USA. And then Waxman and his laboratory staff discovered the several antibiotics including actinomycin, clevacin, streptomycin, streptomycin, glycine, neomycin, and different type of antibiotics they discovered. Streptomycin was the first antibiotics cured the tuberculosis. We know that TB is the deadly disease, and if you look at the history, lots of well-known people, they were killed due to the TB infection. So in 1952, Waxman was the recipient of the Nobel Prize in Physiology or Medicine. It is on the World Health Organization list of essential medicine, and the most effective and safe medicine needed in the health system. So this is the specialty of the Streptomycin. This is very effective against the tuberculosis, and as the WHO, they consider this is a very safe medicine for this. Now let us see that how this antibiotics works. That Streptomycin is a protein synthesizer inhibitor, that essential protein that required for the growth of the bacteria, that synthesis they inhibit. And how it inhibits? Because it binds with the small 16S rRNA of the 30S subunit of the bacterial ribosomes, interfering with the binding of formyl methionine tRNA to 30S subunit. This is how they are binding here, and this lead to complete or partially inhibit the protein synthesis, eventually the death of the cells. Humans have the ribosomes which are structurally different from those in bacteria. So, drug does not have this effect in the human cells. So, human cells that this does not have this effect, this kind of biochemical activity that we do not have the human system, but this is only applicable to the bacterial system. They disturb the essential some protein synthesis, and they kill the microorganisms. Now, if you look at the biosynthesis pathway, the glucose, it produces the glucose 6 phosphate, then it produces that isoacetol, this 1 phosphate, and then D-glucose 1 phosphate to glucosamine 6 phosphate, then myoinositol. This all when they combine, and ultimately they form the dihydrostreptomycin 6 phosphate, and then it ultimately comes finally the streptomycin production. Now, this streptomycin required 30 enzymatic steps, because the several steps are required before it produces streptomycin. Glucose 6 phosphate takes 3 independent route to respectively produce the streptidine 6 phosphate and L-dihydrostreptose and N-methyl glucosamine. The former two compounds condense to form a intermediate which layer combines with methyl glucosamine to produce dihydrostreptomycin 6 phosphate, and this compound in the next couple of reaction gets converted to streptomycin. This is how the streptomycin is produced biochemically. Now, industrial production of streptomycin is like this. It is produced through the submerged fermentation process. I already explained in case of penicillin fermentation process that we use two type of fermentation, not only penicillin in citric acid, also mostly I discussed in citric acid fermentation process that it is usually produced both by 20% citric acid is produced by surface fermentation and 80% by submerged fermentation process. And mostly what we use in the fermentation unit submerged fermentation, the reason is that whole fermentation broth we get the your microorganism will grow and give the product, and that is why it is very effective. So, submerged fermentation is is used here. It is the secondary metabolites, and spores are maintained as soil stock or lyophilized and are used for inoculations, and this is then transferred to germinator where the biomass is increased for inoculating the fermenter. So, here also it is same as we shall have to produce the inoculum before we transfer, we use this in the production fermenter, and this is like this. You have first log phase, you can dioxic type of growth, you can see it here that it grows dioxic growth. We know that dioxic growth means in the it is possible when your organism consume one particular carbon source and then when it is exhausted, then only then the other carbon source will be utilized, then it is called dioxic growth. This usually it has been found that streptomycin fermentation process is follow the dioxic growth pattern. Now, this under the microscope it is very interesting. It has some resemblance with the fungal fermentation, though it is a bacterial fermentation process, but it has some resemblance, because you see in case of vegetative cells they have lot of branches. We can see it. This is what we have in case of fungal fermentation process. The bacterial fermentation, this Streptomyces griseus also have this kind of lot of branches, and also it has this kind of spore formation that is quite common in case of fungal fermentation process. So, lyophilized spore culture are inoculated into soya flower agar media, and they are inoculated at 27 degrees centigrade for 2 to 3 weeks. The spores are then transferred in the sec flux, and following the biomass accumulation and inoculation is done in a sterile media, a concentration 5 to 10%. I told you, whenever we use any kind of inoculum to the production fermenter, we always use 5 to 10% of the volume by volume of the production fermenter. I told you that if the production fermenter size is 100 cubic meter, then 5% is about 5 cubic meter and 10% is about 10 cubic meter. The volume of inoculum for this fermentation process is about 5 to 10 cubic meter. So, usually what we do, we take 90 cubic meter of the media and 5 to 10 cubic meter of the inoculum we inoculate to make the volume of 100 cubic meter. Now, this is the industrial media formulation for streptomycin production. We have two media, one is woodruff and magdenial, and another is the Hokenhull, this media. Then this media contains the soybean milk, but it does not contain, other media does not contain soybean milk, but it has the extracted soybean milk. So, this is total soybean milk that is here used here, and distilled dry soluble, that is also, and pH we maintain here 7.3 to 7.5. Glucose, ammonia and phosphate high quantity inhibit the streptomycin production. Now, if you look at this fermentation requirement, media consists of carbon source like glucose, starch and dextrin, nitrogen source, natural occurring agricultural substances such as soybean, corn step liquor, cotton seed meal, casein hydrolysate. I told you casein hydrolysate mean casein is a kind of protein. When casein we hydrolyze with the help of some kind of protease enzyme, it degraded to the different different size of protein, or it may produce some kind of free amino acid also. So, that is kind of soluble material, because when big protein molecule degrade to the smaller protein molecule, it is becoming soluble, and that is why we call it casein hydrolysate. Yeast and its extract, etc., inorganic substances such as the ammonium sulfate and ammonium phosphate. The vegetable and animal fats like soybean oil, then linseed oil and lard oil that is used, the fatty acid having more than 14 carbon chain length. Because I told you one common thing that we have in the macrobial fermentation process is the foam formation, and particularly when we have aerobic fermentation, we have a foam formation, and during foam formation I told you that this is it will keep on rising, and if you enter into the mechanical seal, your system will be contaminated, and then the whole fermentation process will suffer. You have to discard the whole fermentation broth. So, this is an important thing that everybody has to consider, that how to subside this foam. And I told you before also, the different fermentation process we use the different antifoam oil, because we shall have to choose the antifoam oil on the fact that the kind of antifoam oil that we use, that should not affect the fermentation process. Now, in case of streptomycin fermentation process, they found the soybean oil, linseed oil and lard oil, which has for more than 14 carbon chain, they are suitable for this as the antifoam oil. Now, fermentation conditions at the optimum temperature 28 degrees centigrade, pH range is 7.6 to 8, high agitation and aeration are required. The fermentation times 5 to 7 days, aeration is 0.5 to 1 ppm, This I want to point out that VVM means, I hope you might be knowing, VVM means volume of air per volume of liquid per minute. So, it is like this. Suppose we have this is the fermenter and the working volume, let us assume this we are doing the aeration herein. So, let us assume this working volume is 10 liter and if you say this is 1 ppm, that means 1 liter, 1 liter the 1 ppm means 10 liter, if you 1 ppm 10 liter per 10 liter volume of the medium per minute. So, this is like this, this is how the aeration is considered. So, we will have to the flow rate of this here, the flow rate will be 10 liter per minutes. So, since it is capacity 10 liters, so VVM will be 1 VVM is like this. Inadequate supply of oxygen leads to accumulation of pyruvate and lactate, that is also very important factor. The phase involved in the streptomycin fermentation, several phases are there. That phase 1, it lasts for 24 hours, pH increases to 7.5. This highly active, the proteolytic property of the organism set free the ammonia that into the media of the swamil, and this due to the formation of the ammonia, the pH rises. Streptomycin is produced less quantity at this phase and rapid growth occurs to produce mycelial biomass, and we have already shown, though it is the bacterial cells, but the growth characteristics is a little bit similar with the fungal cells. However, glucose uptake is low. Now, in case of phase 2, it is most critical. Rapid production of streptomycin is observed, extend for 1 day to 5 to 6 days under sterile conditions, and then very less growth of biomass is observed. Ammonia is consumed and weight of the mycelia remain constant, because this is also a secondary metabolite production, because this also take place at the stationary phase of the fermentation. The phase involved that this phase involve the high glucose uptake, but phase 1 I have shown the glucose uptake is less, but here is very high, pH increases to 8, and in phase 3 the sugar depletion occurs and then antibiotics production ceases, the harvesting is done and pH remain fairly constant between 7.6 and 9. Now, if you look at the different profiles of the streptomycin production, it looks like this. Now, here it is very the important thing is the glucose. If you look at this is the carbon source, it will keep on decreasing like this, and this is in the fermentation time in days. So, after 6 days of fermentation, most of the glucose will be exhausted. Now, if you look at the mycelium or cell mass production, if you keep on increasing, and then it is biphasic type, it increases like this, then it increases like this, then it decreases like this. So, it is like this. Then pH, if you look at pH is keep on steadily increases. Initially it will be at a very slow rate, but finally it increases quite high. And here is the streptomycin production is when this organism almost come here, it will produce like this, streptomycin production take place and slowly it increases and then attains the plateau, then again it decreases like this. So, maximum you can say the maximum streptomycin production that take place on 7 days of fermentation. On 7 days of fermentation we get the maximum streptomycin production. Now, streptomycin recovery also very important aspects, because how the streptomycin recovery takes place. Now, this is usually take place by the adsorption phenomena. Now, how it is done? We have the filtered broth, then we separate the cell, maybe either centrifugation, mostly we use by the cell separate by centrifugation process. Then we have the dilute broth. We pass through this amber light, this ICR, sodium form ion with the cation exchange column, we used to pass that. Then your streptomycin get adsorbed on the surface. Then we remove the ions by washing with EDTA at pH 8. Then finally, we do the elution by 2.5 normal H2SO4 until the pH drop to 5, and then we get the liquid. That liquid we decolorize by using with the DAR-SO G6 column. Then antigen removal by filtration with the polyacrylamide gel, cellulose acetate, or by dialysis. Cellulose acetate, you know that this is the kind of membrane that we have. And then finally, we do the concentration by evaporation techniques, and after evaporation we get this powder, white powder in the form of the streptomycin sulfate. So, we get the streptomycin in the form of streptomycin sulfate. The recovery of streptomycin is that micellar is separated from the broth by filtration. Then streptomycin and other amino glycosides are basic in nature. The remaining liquid is then percolated through the cation exchange resin column, where the streptomycin gets absorbed. Then it is finally eluted by washing with buffer, and further impurities are removed by treating with sodium hypochlorite or EDTA or activated carbon, because activated carbon I told you before also largely used by chemical and biochemical industry as a decolorizing agent. And then streptomycin can be precipitated in the form of sulfate. When it combines with the H2SO4, it forms the calcium streptomycin sulfate with soluble, and the purified streptomycin sulfate solution is concentrated under vacuum and dried, and dried it aseptically, and finally the product is tested and purity and packaged. So, this is how the streptomycin production take place, because let me conclude here that streptomycin is kind of antibiotic that is produced through the bacterial fermentation process. Most of the antibiotics is produced through the fungal fermentation process. Couple of antibiotics they can only produce through the bacterial fermentation. Streptomyces griseus, whose nature is little bit similar as compared to the fungal cell, because it like fungal cell, it has the sporulation characteristics, and also this Streptomyces griseus, it has also sporulation characteristics, and when it grow in the liquid, it gives the that mycelial production, that same as in case of fungal cells. So, and this the streptomycin also considered as the secondary metabolites that produce, and it takes quite long time, like similar to penicillin fermentation process. The penicillin and streptomycin, the cephalosporin, it 6 to 7 days fermentation process is taking place. After the fermentation is over, we separate the cells by through the filtration process, and since if the size of the cells is big, we can use the rotary vacuum filter for separating the cells, and size is small, then we use the centrifugation technique, and then we pass it through this absorption column. Adsorption column they absorb the streptomycin, and then after adsorption we elute with it is absorbed in the cation exchange membrane, and then we elute by using the 2.5 normal of H2SO4 so that streptomycin sulfate will go out. Then we do little bit of purification, we remove the impurities that present in this particular the elute, that we color we remove, and then finally we dry it by using the vacuum dryer. As we know that that at a low vacuum, the boiling point of the liquid, that water decreases. So you can if you do the vacuum drying techniques, then you can preserve the quality of the media. And this whole operation is usually done under aseptic condition, because it is used for the therapeutic purpose. So this is all about the antibiotics fermentation process I want to cover, and in the next class, next lecture, I try to discuss one very interesting fermentation process that is used by the industry, that is the Baker's yeast fermentation process. Thank you very much.